Welcome to this CoBCoM lecture, and today we are going to talk about brain network alignment and similarity. I am Matteo Frigo, and I did this work together with Emilio Cruciani, David Couder, Rachid Deriche, Emanuele Natale, and Samuel Delory-Gauthier. We are all affiliated to Inria Sophia Antipolis Méditerranée at the University of Côte d'Azur in France, and we belong to two different teams, the Athena project team and the COATI project team. We have two different core interests that intersected in this work, in this joint work that we recently published in Network Neuroscience, where we explored the problem of network alignment and similarity, showing how these two concepts reveal atlas-based topological differences in structural connectomes. So in the next minutes, we are going to divide the talk in three parts. The first part, which is going to be presented by me, is going to be about the context a bit and the research question that we are going to ask ourselves. In particular, I will introduce the concepts of brain structural connectivity, the concept of atlas of the human brain, and I will introduce the necessary tools to understand the research questions, which concerns the link between atlas of the human brain and topology of the brain network. Following this, we will have Emilio Cruciani, which is going to give us the more theoretical flavored part of this talk concerning the methodology, which includes two main parts. The first one concerns the network similarity problem, in which he will introduce the graph Jaccard index, and then he will introduce us the WL-align algorithm for the graph alignment problem. Finally, Samuel Delory-Gauthier is going to show us the results of our work and discuss them, and in particular showing the experimental setup, which uses diffusion MRI data from the Human Connectome project, and showing us how the graph Jaccard index and the WL-align algorithm together, they give us useful criteria for choosing an atlas that is suitable for structural connectivity studies. All the codes and data that have been used in this work are available online. You can find most of them at the repository at the DOI of the paper, but the WL-align algorithm is now contained also into a Python package that is publicly available at the link that you see here. So let's start from part one that I am going to take over, and here we introduce a bit the context of the work. The human brain is a complex multi-scale biomechanism where microscopic interactions have macroscopic effects and vice versa. And also it is a very subject-specific brain. We cannot just study one brain. We have to study all our brains. Also, the brain changes over time with learning, development, evolution, and aging and pathologies. And it is characterized by a very high dimensionality because we are talking about something like 80 billion neurons, which are connected by around 160 kilometer myelinated axons. Myelinated axons are one part of the neuronal cells that you can see here on the top right of the slide. And the myelinated axons connect what is the nucleus and the soma of the neuronal cells with the axonal terminations. And they somehow transport the activation from one side of the other of these elongated cells. So we have learned from studies in neuroanatomy that the axons, the myelinated axons, compose the so-called white matter of the brain, while the nucleus, soma, dendrites, and the axonal terminations, therefore the two sides, the two parts that are on the sides of the axons, they form the gray matter. Gray matter is what composes the cortex of the brain, therefore the part that is on the outside of the human brain. And each region of the gray matter, and therefore of the cerebral cortex, is devoted to a specific function of the brain. For instance, we know that the precentral gyrus is devoted to the primary motor tasks, or the postcentral gyrus are devoted to primary sensory tasks. Or we have other regions that are devoted to auditory or understanding speech or visual association tasks. Anyway, the cortex is what, the cortex and therefore the gray matter is what is associated to the functioning of the brain. And the dual concept to this is what lies inside the brain, which is the white matter. And the white matter, on the contrary, is made of connections, which are represented by the axons that we saw before in the structure of the neuron. And these axons are basically the trajectories that are followed by the activations that are transported from one region of the cortex to another. With tractography, we're able to draw these three-dimensional trajectories that are followed by axons, and therefore that connect these different regions of the cortex. And we will see later how we can do that with diffusion MRI. So in order to describe these concepts, these dual concepts of the regions that talk to each other by means of the regions that lie in the gray matter, and they talk to each other by means of connections that lie in the white matter, in 2005, Patrick Ackman and Olaf Sporns introduced the concept of connectome, which is a graph-like representation of the brain network where nodes are regions of the gray matter and edges are connections between these brain regions. We have to do a step back here because in principle, we would need to talk of neuron against neuron interactions. But as we said before, we have around 80 billion neurons, which makes this problem intractable from the practical point of view. Therefore, we need some simplifying assumption. And in particular, the simplifying assumptions that we employ when we study connectomes are that neurons are grouped in coherent ways. In particular, we use atlases of the cortex that subdivide regions of the cortex into coherent regions that either lie on specific locations or are associated to the same functions or that respect a mix of these criteria. As you can see on the top right of this slide, we can have either binary connectomes, so binary graphs, or we have the concept of weighted graphs, weighted connectomes. These two encode some complementary information. In particular, the weighted connectome extends the concept of binary connectome by considering the connection strength between different brain regions. What we do when we study this type of connectivity is to study structural connectivity, where we estimate the strength of the axonal connectivity between regions. Imagine that our cortex is subdivided into 4 regions, A, B, C, and D, and that we were able to track some connections that are here drawn as red lines. In order to define a structural connectome out of this description, we just have to look at how each pair of region is connected by these lines. For instance, regions A and B are connected by only one streamline, one axonal pathway. Therefore, we will encode in our connectome an edge between regions A and B that contains a weight that is proportional to this number of axonal pathways that we encoded, that we recorded in our estimation in our tractography process. And therefore, here's the one that we have in the adjacency matrix or connectivity matrix that we can define for this brain graph. If we repeat this process for each pair of regions, we can recover all the other edge weights between regions. And notice that these edge weights will be all positive and the graph will be undirected. Why this? Because basically because with the techniques that we have at our disposal do not allow us to distinguish afferent and efferent connections in structural connectivity studies. And moreover, the weights are all positive because basically we are associating to each edge some connectivity strength. We cannot have a negative strength of an edge of a connection, of an axonal connection. And in the slides before, we saw how we can define the edges of a brain graph, but how to define the nodes? Well, the atlases of the human brain that we can employ are of several kinds. We have here 4 different examples. One is the Desikan-Kileni atlas that subdivides the cortex into 68 regions that are drawn following anatomical criteria. Otherwise, we have on the bottom left the Jaccard structural connectivity, structure-based parcellation, which can subdivide the cortex in n regions with n an arbitrary number. And these regions are delineated following the structural connectivity patterns within the cortex. The somehow corresponding atlas in the functional domain is the Schaeffer atlas, which allows to delineate the regions that talk to each other in coherent way from the functional point of view. And finally, we have the Glasser atlas on the top right that is delineated following a mixture of all these criteria, therefore anatomical, structural, and functional, and that subdivides the cortex into 360 regions. Choosing an atlas is not a trivial task to do, because sometimes we need specific data to compute specific parcellations. And also, usually, the criteria with which we choose an atlas are based also on the availability of the software to compute a certain atlas, whether this software is free, I can use it basically, or if it is fast and it can be used in a reasonable time. Moreover, we always tend to use atlases that have been used in similar studies in the domain that we are conducting our experiment. Also, the atlas that we are going to use in a study must provide interpretable insights into the specific domain that we are embedding our setting, that we are embedding our experiment in. Finally, the subject must be used for the atlas must be used for inter-subject comparisons, which makes it even more, and this criteria makes it even more selective to choose a parcellation. But in general, as you can see from these 5 points, we don't really have a quantitative criteria that allows us to choose an atlas in a proper way. So an additional note on the choice of these atlases is that we have basically 2 types of parcellations. We have group or template-based parcellations, where basically we have the same template that is projected on the subject-specific spaces. And this type of parcellation reflects somehow the average subdivision of the cortex that can be found in the studied cohort. And this type of parcellation allows to have bijective correspondences between the regions in different subjects. On the contrary, when we use subject-specific parcellations, we have a better reflection of the underlying subdivision of the cortex, because we are embedding everything into the subject-specific space, but we have no a priori one-to-one correspondence between these regions. Therefore, the comparison between the results obtained on different subjects might be more tricky. So the question here is, given the plethora of atlases that can we choose, how does the choice of the atlas affect the topology of the corresponding structural connectome? The question that we asked in the previous slide is here embedded into a more specific framework, which is that of, given an atlas, how robustly does it capture the topology of the structural connectome? And we try to answer this question by taking as a granted assumption that brain networks obtained from a homogeneous cohort of subjects have a highly similar topology. And we test this assumption with a robustness metric, which is given by how simple it is to compute a graph alignment between 2 brain networks of 2 different subjects. And by graph alignment, we mean to find a map between the nodes of 2 different, of the connectomes of 2 different subjects by looking uniquely at the edges of the 2 connectomes. So we separate the planes of the nodes and the edges, and we look only at the edges to find correspondences between the nodes.
So now let's move to the methods part. As we said before, brain networks are represented as edge-weighted graphs. Nodes of the graphs represent the regions of the brains according to some atlas, for example. Edges represent the connection between such regions, and weights instead the strength of such connections. The graphs are represented as weighted adjacency matrices, in which the entry in the element in position UV is equal to the weight of the strength of the connection between areas U and V. In this paper, we introduce a new measure of network similarity, which is called the Graph Jaccard Similarity Index, or GJI. Our point of view, differently from other metrics that are already used in the literature, is that we want to generalize the Jaccard index, which is a similarity measure for sets, to the setting of graphs. Jaccard index, as you can see in the example, measures the similarity between two sets, A and B, and is defined as follows. At the numerator, we have the size of the intersection between the two sets, and at the denominator, we have the size of the union. So looking at the example here, we have set A with red dots, set B with blue dots, and the Jaccard ratio, the Jaccard index, is defined as 3, which is the size of the intersection, divided by 33, which is the size of the union. Of course, if the two sets A and B are disjoint, so they do not overlap, the Jaccard similarity will be 0. And instead, if the two sets coincide, the Jaccard similarity is 1. What we do in our paper is to extend this notion of similarity between sets to similarity between weighted sets, and in particular, weighted graphs. So given the two graphs X and Y, where X and Y are actually the adjacency matrices of the graphs, we define the Jaccard similarity index, the graph Jaccard similarity index, as the ratio between the sum over all possible edges in the graph of the minimum between the two elements, divided by the sum, again, over all the possible edges of the graph, of the maximum between the two corresponding elements. In particular, this measure generalizes the Jaccard index for sets, because if you consider the graphs with no weights, so the entry of the matrix are just 0 or 1, this exact formulation is the same as the Jaccard index. Looking at the example here, we have X and Y, the two graphs, X in red and Y in blue. And to compute their graph Jaccard similarity, what we do is consider the minimum graph and the maximum graph, respectively, in the yellow and green box. Each edge, so for example, the edge between node A and B, in the minimum graph is the minimum between the weights of the edge between A and B in the two graphs. So for example, here we have 0 because AB, the edge AB in graph X weighs 1, while there is no edge between A and B in graph Y. So the weight is equivalent to 0. And we do the same for all the edges in the graph, and we apply the formula. And this number tells us the amount of similarity between the two graphs. What is nice about this new similarity measure is that it generalizes something which is very used as the Jaccard index, for example, in data mining and many other areas. It can be used to and can be applied for the graph isomorphism problem, which, as we will see in the next slides, is very related to the graph alignment problem. It has a very simple interpretation, and moreover, if we consider the complement to 1 of the metric of the measure, it induces a real metric. So as mentioned before, there exist other similarity measures between graphs, and in particular, given two graphs, G1 and G2, one possible way to measure their similarity is the correlation, which is nothing else than the cosine of the angle between the vector representation of the two adjacency matrices. And indeed, it is computed as the scalar product between the two vectors divided by the product of the two norms. And this is a measure of similarity, since the closer, the smaller is the cosine, and the closer the two vectors are to be parallel, actually one over the other. Instead, a possible way of measuring distance between two networks is that of considering the norm of the difference between the two matrices. And one of the most used is the Frobenius norm, which considers, which is essentially the two norm while considering matrices as vectors. Okay. Of course, if the matrices are equal, the norm of the difference will be 0. Instead, the farther they are from one from the other, the higher will be their distance. In the paper, we compare the graph Jaccard index with these other similarity matrices measures. So let's move to the graph alignment problem. As said before, we are given two graphs, G1 and G2, and we want to find a matching that goes from the nodes of V1 to the nodes of V2. Of course, intuitively, what we want that the matching, the alignment between the two graphs maximizes some notion of similarity between the two graphs. Okay, and we can note that the graph alignment problem is a generalization of the graph isomorphism. Indeed, if the input graphs G1 and G2 are the same graph, but just permutated according to some random permutation, the optimal alignment is the isomorphism between the two graphs. And in particular, this notion of graph isomorphism is very important in our setting, because we leverage it to introduce a new alignment algorithm. So graph isomorphism is not an easy problem. Indeed, there is no known polynomial algorithm to solve it, but it is not even known if the problem is in the NP-complete class. The Weisfeiler-Leman non-isomorphism test is a procedure that tells you if two graphs are not isomorphic. In particular, the procedure assigns a signature to every node of the graph. And if the signatures do not match, the procedure tells you, guarantees you that the two graphs are not isomorphic. Otherwise, if as in this example, so these two graphs are isomorphic, the procedure will output compatible signatures. For example, orange node here has 9 and the other has 9, 8 and 8, and so on. But this doesn't give you any guarantee, because also non-isomorphic graph could produce the same signature. In our setting, what we want to do is to adapt this procedure, the Weisfeiler-Leman, to the case in which we don't want to check for equality, but just for similarity. Okay, so we propose this extension of the algorithm that takes two parameters, k and l, which are parameters of a visit of the graph. And yes, the algorithm works as follows. So given an initial node u of the graph, we do essentially a breadth-first search starting from u of width k and depth l. So we will go deep for L hopes starting from U, and in each hope we will just consider K nodes. In the example, we'll consider K and L equal to 2, just for simplicity. So starting from node U, we look at the first, we want to produce a signature corresponding to the node. The first component of the signature is just the volume of node U, where the volume is the sum of the weights outcoming from node U. Okay. Then for components 2 and 3, and there are 2 because the width of the parameter width is 2. What we do is the following. We consider all the possible paths outcoming from U of length 1. So UV1, UV2, and UV3. And for each of them, we compute this function f, which is a scoring function that essentially promotes nodes which have a high strength, so a higher correlation between U and its outcoming neighbor. Okay. Note that the function penalizes long paths, since it is essentially the product of the weights normalized by the volume of each edge of a path that we are considering. So in this example, starting from U, we assume that f of u v1 is greater than f of u v2 greater than all the others. And what we consider is just the biggest two of the biggest k in general of these functions. Okay, so in particular, they're going to be v1 and v2. So the next two components of the signature, components 2 and 3, will be equivalent to f of this function f of the path that goes from u to v1 as in second place and u to v2 in third place. After that, we continue the breadth first search. So starting from v1, we go another level deep, and in particular here in the bottom, we look at the blue circle. And again, from v1, we have w1, w2, w3, and so on. And we assume that, sorry, and also the back edge u. So in particular, we can assume that the function f of u, v1, u, so u, v1, and then going back to u, is bigger than f of u, v1, w1. So f of u, v1, w1. We compute these two functions and use them as next in the signature. And we do the same for the other child of u, so v2. Once we have the signatures, we can explain how the actual WL-align algorithm works. So essentially, given the two input graphs, we compute a signature for each node of the graph. After that, we set up a bipartite graph where nodes in each partition represents the nodes of each network. And this bipartite graph is complete. And in each edge, we set a weight which is equal to the Euclidean distance between the two signatures, which are just vectors in the space. After we have this bipartite graph set up, we run the Hungarian algorithm that finds an alignment M, which is just the minimum weight bipartite matching, taking as input such a bipartite graph. In the paper, we compare the WL-align algorithm with other state-of-the-art algorithms, and in particular with the FAQ, the fast approximate quadratic programming for graph matching. So this algorithm is just an optimization algorithm that essentially maximizes the Frobenius norm of the difference between G1 and G2, so the two input graphs, but where G2 is permuted according to the solution P of the problem. And in a nutshell, it works as follows. So we start from an arbitrary B stochastic matrix, which is just a relaxation of the permutation matrix P that we want to find. After that, it iteratively follows the gradient and looks for a local solution of the relaxed quadratic assignment problem, which is a sort of dual version of the graph matching problem. And after having found the local solution, it projects it back to the domain of permutation matrices.
Okay, thank you, Emilio. And I'll be discussing the results of our algorithm. So our experimental data consisted of 100 unrelated subjects from the Human Connectome project. We used the minimally processed diffusion data, and for each subject, we computed the fibro-DFs using multi-shell, multi-tissue constrained spherical deconvolution. We then produced connectomes using anatomically constrained probabilistic tractography with 5 million streamlines per subject. We used streamline counting to produce normalized streamline count connectomes using the different atlases. So we used 4 atlases: the SICAN atlas, which is computed using FreeSurfer, the Glasser atlas, which is provided directly on the HCP subjects, the Guicciardo atlas, which is based on anatomical connectivity, and we used 11 different resolutions ranging from 50 to 1,000 regions, and the Schaefer atlas, which is based on functional connectivity, and we also used 10 resolutions ranging from 100 to 1,000 atlases. So you see on the bottom of the screen examples of these atlases for some random coloring of the parcels. So the Desikan Atlas, which has 70 regions per hemisphere, the Glasser Atlas, which has 180, Jaccard, in this case, with 100 parcels, and the Schaefer Atlas with 800 in this case. The objective of our experiments was to compare the performance of two different tools, the WL-Align, which provides network alignment, and the Graph Jaccard Index, which measures the similarity between networks. To do this, for each atlas, we had a ground truth matching, which we'll call M star, between subject, which is basically how a subject matches with himself. For each pair of subject, we can also compute a different alignment, either with WL-Align or FAQ, for using the parameters k equals 2 and L equals log m. We use different metrics to measure the performance, so some of them were described earlier. So the Graph Jaccard Index has already been presented. There's also the Graph Jaccard Index ratio, which we'll call just J ratio. And it is defined as the Jaccard Index over the Jaccard Index of the ground truth matching, the M star that I just discussed. There's also the node matching ratio, which is how a node is aligned against itself, and the Frobenius norm, which was discussed previously. Before moving on to the performance of the WL-Align algorithm, let's first compare the different similarities and distances that we introduced. So what we see here is the Graph Jaccard Index, the Frobenius norm, and the correlation for each subject of the database with all other subjects. What we can observe, the different trends, is that as we increase the number of parcels, both the Jaccard Index and the correlation decrease, meaning the problem becomes harder to solve. This is simply because the underlying graph alignment problem becomes more difficult as the number of nodes increases. Notice that the Frobenius norm, the y-axis is inverted, and so we observe the reverse trend. As the number of parcels increases, the Frobenius norm diminishes. So the technical reason why this happens is discussed in more detail in the paper. In summary, atlases with few parcels have a higher matching ratio and a higher Jaccard Index than atlases with many parcels. We also observed that the atlases that are based on structural connectivity perform better than the atlases which are based on functional connectivity. And this is normal because our matching in this particular instance is based on structural connectivity. We also observed that the Glasser Atlas, which uses both functional and structural connectivity, lies somewhere in between the Jaccard Atlas, which is based purely on structural connectivity, and the Schaefer Atlas, which is based purely on functional connectivity. An interesting fact is that the Desikan Atlas outperforms all other atlases for both the Jaccard Index and the node matching ratio. And in all our experiments, WL-Align outperformed the FAQ algorithm. We also observed that the Frobenius norm, because of its inverted trend, is inadequate to compare atlases. Illustrating here the percentage of correct node matching directly on the brain, we can have a summary of the results that we observed. So for the atlases with a low number of parcellation, for example, the Jaccard 50 and the Desikan Atlas, we can see that the self-node matching, which is basically how many times a particular region was matched against itself, has no real spatial correlation. In this case, in our experiment, each hemisphere was matched independently, and we can see that even across hemisphere, the easier to match regions do not correspond across hemispheres. We can also observe that as we increase the number of regions, the number of self-matching rapidly decreases. And so for the Jaccard 1000 and Schaefer 1000, it is very rare for a region to be matched against itself. This is, again, normal because as we increase the number of regions, the number of nodes increases and the graph matching problem becomes much more difficult. In summary, in this talk, we discussed graph alignment for brain networks. Graph alignment allows us to use subject-specific parcellations while preserving correspondence across subjects. We also introduced a new similarity index, which is the Graph Jaccard Similarity Index, a generalization of the similarity for less structured data, such as sets. It's an index that applies to the graph isomorphism problem, and it has a simple interpretation, 0 being a very poor similarity and 1 being perfect matching. It also induces a metric unlike the correlation. We also introduced a new alignment algorithm, WL-Align, which allowed us to gain insight onto how atlases influence the topology of structural networks. So again, I remind that all of these results are discussed in detail in our new paper in Network Neuroscience, and you have the reference here. All of the code that was used to generate the figures and the results is also available publicly as a Python package with the link illustrated here. This work was funded by the European Research Council under the European Union's Horizon 2020 Research and Innovation Program under the grant ERC CoBCoM Computational Brain Connectivity Mapping. Thank you.